Hi everybody. I hope that you are all doing well. I have had lots of conversations about practice problems, which is awesome, and please let me know if you have further questions. Remember that we're going to have a review session tomorrow at 7. I sent you guys the Zoom link, and I also made a calendar invite since I was putting it on my calendar anyway. I figured, why not? And the Zoom link is in the calendar invite as well. So that's that. Remember that you're going to have, obviously, the exam on Friday. Sorry if you didn't know that until right now. I will get you the key to the T-cell development problem set's already posted. I will get you your graded versions ASAP. So today we're going to be talking about T-cell effector function. And so just to kind of remind you where we're at, what's been going on, in a lot of ways, I think of sort of this section that we've been thinking about recently as kind of this question of what the heck takes so long, adaptive immune system? Why is it that we go from our infection to really having this nice measurable response almost a week later? What takes so long? What are the events in this situation? Note that I said measurable, and I guess the answer is easily measurable because, in fact, yes, you do have like the one cell that resulted from VDJ recombination even before this. It's waiting until you actually have many of those cells that have undergone proliferation that you can actually easily measure them. In the past, we couldn't. Now we can, but it's hard. And you can see I showed you some data for people who really want exact numbers in days on the right. And we've seen that some of the issues are that we have to get our microbe, our antigen to the secondary lymphoid organ. We have to get our naive B and T cell to the secondary lymphoid organ. So we need the trafficking stuff. We need the T cell to interact with the antigen presenting cell, and we need antigen presentation to occur. And so you can see that happening here. That T cell has to get activated through the steps we've been talking about with signal 1 and signal 2. Needs to make IL-2 as well as respond to IL-2. And do some cell division, and it also needs to differentiate or otherwise sort of gain new effector functions, gain new functions, so that it can now deal with pathogens. And so really what we're going to be thinking about a lot today is sort of what the result of that differentiation looks like, what's going on with that cell after it has differentiated. That cell, after it does, this clonal expansion, this division that's happening because of IL-2 is still happening in the lymph node. This is why your lymph nodes get swollen when you have an infection. It's because they're getting full because of all of these proliferating cells. That's why they're swelling, is because they're just getting slam packed with all these cells that are dividing. And then those cells are going to, as you can see, sort of move to whatever tissue, whatever peripheral tissue outside of the lymph node is the site where you have the infection with the pathogen, where now we're going to see that effector function, that function that the cell kind of learned how to do as a result of its activation. That's now going to be happening to help us get rid of the pathogen, and that's what we're seeing today. Jazz, you had a question? In all of the experiments and like the data that I was showing you, I was taking blood samples. But again, if you're trying to look at rare cells, that can be really hard. And so we have fancy techniques now that can let you do I'm blanking on the word, like amplification, or like to really zoom in on those rare cells. Those are relatively new types of techniques. So when immunologists sort of started looking at this, you couldn't get those rare ones at the beginning. Now you can. And so that's part of why it looks like the answer is 0 at the beginning, and it's because it was actually just really hard to measure them for a long time. Whereas here we finally have gotten that cell division to happen, and we actually have enough cells that we can see them. So does that sort of answer? Okay. Yeah. So we're going to talk about this differentiation process, or the details of this differentiation, for first our CD8s and then for our CD4s. And so to start out with, sort of starting this process, our CD8 cell is a naive CD8 cell. When we start the process with the CD4 cell, it will be a naive CD4 cell. And eventually that cell, like I said, is going to sort of gain some functions. It's going to have been changed by the experience of having seen the antigen and now learn how to do something. And so once our CD8 T cell is activated, it is no longer called a naive CD8 T cell. It is now called a cytotoxic T cell, sometimes also referred to as a CTL. And now this cell has this ability to be cytotoxic. CTL stands for cytotoxic T lymphocyte, which means it can kill other cells that are infected. And so you can see this sort of process happening here. Our naive CD8 T cell will see antigen. It will get signal 1 and signal 2. And it will start to divide. And so instead of there being one CD8 T cell, there will be a lot of CD8 T cells at that location. This is still in the lymph node. Our lymph node is swollen because there's so many cells. It also might interact with CD4s. I went back and forth about whether to block off this part of the slide. Yeah, it could also interact with CD4s. The reason why I decided not to is just because I do like the fact that it shows that multiple T cells could be interacting with the same dendritic cell and they could share cytokines. But I don't care that it's a CD4 right now. At that first interaction, the first time the T cell is seeing, say, the dendritic cell, that it's a naive CD8 T cell, it doesn't know how to kill yet. After it has gone through these divisions, it is going to be activated, and it's now going to be this type of cell called a cytotoxic T lymphocyte, or a CTL. That is going to be able to kill infected cells in the tissue. And so this CTL now, so here it is, the first time interacting with the dendritic cell getting activated. Once we've activated it, it's going to go somewhere in the tissue. And now when it sees antigen again, now that it's activated, it will kill that cell that was presenting the antigen. So it didn't kill the original dendritic cell. The original dendritic cell turned it on, told that you should grow up and become a CTL now. Once it has grown up and become a CTL, now when it's going to see its antigen on MHC, class 1, then it's going to actually kill that infected cell. And so it's the effector function of the CTL is to kill other cell types. I had a video that doesn't work anymore, so I went and found a separate video, which is this one that is linked here, that I might even be able to show you. I don't know if I know how to make it full screen. But in any case, in purple, you are seeing a cytotoxic T cell, a CTL. In blue, you are seeing what we will just call an infected cell. It's basically the cell to be killed. Yes, it is technically a cancer cell in this case, but we're not going to deal with that right now. We're just going to think of that purple or the blue cell as the effector cell. You're going to think of the CTL as here, so you can see this process. So basically what you see is that the CTL is going to secrete something. You can see, if I replay it, the CTL basically gives something into this cancer cell, and this cancer cell then undergoes apoptosis, or this infected cell undergoes apoptosis. And so that's what it looks like when a CTL is killing an infected cell. Yep.
So when you said it's trying to be activated,
Mm-hmm.
basically 1 and 2, that's just a CD8 T cell, and then once it only needs signal 1 is a CTL.
Yeah, once it's been activated, it's a CTL. So Oh, that wasn't quite right. This will work. So that little video kind of shows you basically this killing process in real time. And one thing that you should notice from that video is that the infected cell died. The CTL was fine. It just sort of sat there. You saw the one cell undergoing apoptosis, and the CD8 T cell was just kind of chilling. That CD8 T cell can actually go on and find another infected cell and kill another infected cell. So that CD8 T cell is going to interact specifically with one cell, make that cell die, and then it can find another cell and kill that cell, and find another cell and kill that cell. So we might have that one CTL killing multiple cells. One thing that you could see in that video, it's also kind of shown here, is it is a specific interaction. So this cell is going to interact with this other infected cell. And remember how you saw in the video kind of like a little pulse of material get delivered from one cell to the other? It is a specific interaction. This cell has specifically said, I want to kill this one. It's not like it's going to kill everything in the area. So for example, if you have a CTL in your lung trying to kill influenza infected cells, it's going to zero in on exactly the one influenza infected cell and kill just that one. And it's going to leave everything else untouched. So it's not like it's just going to go and kill everything around. It's going to interact specifically, individually with each cell, look at the MHC, and potentially kill that cell individually. It could go on and then kill another cell later that is presenting, but it's not going in sort of 360 degrees, killing in every direction all at once. Yep, Emily.
So even if a CTL has been deactivated with the PD-1 CTLA4, it's still considered a CTL? Or like that terminology?
Why is this being weird? Yeah, I would. Yes. Okay. For your purposes, yes. For the purposes of like my PhD thesis, no, there were way more weeds than that, but I don't need to tell you those weeds, and I'm going to hold myself back from it right now. Okay. So we also need to think a little bit about what kind of molecules or proteins are involved in this specific killing. So what does the CTL use to kill other cells? There are a couple of different answers. One of these answers is that there is a protein on the surface of a CTL called Fas ligand, which can interact with a protein on the surface of the infected cell called Fas. So we can have a Fas ligand interaction. And if this infected cell gets triggered through Fas, that can induce apoptosis. And so one thing that the CTL can do is it can show its Fas ligand to the infected cell, and that will signal the infected cell to undergo apoptosis. So Fas ligand is one way that we get killing from the CTL. Yep, Jazz.
So this interaction happens, and then the CTL gives off those things.
That's going to be on the next slide. So basically the idea is that there are a few different ways CTLs can kill. One of them is Fas ligand. The more famous one is the giving off of the things. So there's sort of alternatives. The CTL probably is doing multiple of them at once. One alternative is this. And so you'll see why I'm going in this order in a second. So here you can see in the pink is the T cell with Fas ligand on its surface. Here in the orange is the infected cell with Fas on the surface. And you can see that there are a bunch of steps of signaling that happen downstream of Fas. I want to point out a couple of steps of this. One, we have some stuff happen that eventually gets us to stuff going on at the mitochondria and all the apoptosis things that you learned about in Bio 250. So there's like a bunch of steps, get us to some mitochondria business. And eventually we turn on caspase 3, which actually kills the cell or leads to apoptosis. So caspase 3 is sometimes known as the executioner caspase because it actually cleaves all the stuff that really kills the cell. And that happens after all these steps of mitochondria stuff. So basically caspase 3 starts out as inactive, binds to some mitochondria stuff, that makes it become active, and that leads to all the things that we see in apoptosis. Okay? So this is one way that a CTL can make a target cell die. The other way that a CTL can make a target cell die is that there are a bunch of granules in that CTL, and it can secrete those granules straight at the target cell. So these are known as cytotoxic granules. So here it says LG or lytic granule. It's the same thing. You've got these granules full of toxic stuff. The CTL will basically secrete those toxic granules right at the infected cell that it's trying to kill. And you can kind of see that happening in the video. One other thing that's kind of nice is that if you remember, I told you when a T cell is interacting with another cell, there are lots of proteins that are kind of stabilizing the interaction. And I mentioned that there were some integrins stabilizing that interaction, and I kind of mentioned that they make like a little wall. So there's like a ring of integrins around this interaction site. The CTL can actually secrete its toxic stuff right there, and the toxic stuff can only go to the infected cell. It can't like leak out and go to random places because there's this integrin wall keeping it in place. So it basically shoots its toxic stuff really directly at the infected cell, kind of in between that integrin wall. And so you can see basically the CTL, part of it being activated, part of it going from a naive CD8 T cell to a CTL is it makes these granules and gets them ready to go. And then as soon as it sees its target, it can then release the granules at the target to kill the target. And so you can see this happening in cartoon form on the left. And you can actually see that here are the granules that are labeled. They're usually just sort of all over the cell. But when the CTL starts interacting with an infected cell, it puts them all right at the border and shoots them right at the infected cell, kind of right at this interaction site. So in this slide, I'm telling you that the stuff that's in those granules is toxic stuff. I'm calling them cytotoxic granules, but I haven't mentioned specifically what is in those granules. The truthful answer is a lot of things. I'm not going to tell you every single thing that's in the granules. I'm going to tell you two famous things that are in the granules. Here you can also see another view of this whole thing. Here's our CTL. It's been activated, so it's got a bunch of granules. It'll interact with the target cell. It'll basically rearrange so all of the granules are next to the target cell. And then it'll shoot the granules right at the target cell to kill it. You can see the toxic stuff is only going to this cell that we want to kill. It's not like going randomly around to like destroy the tissue. Now the CTL is going to leave and the target cell is going to undergo apoptosis. And the CTL is going to be fine. It could go find another target cell and kill it. So you can see this whole process happening here. Yep, Chrissy.
What is the time for the CTL recycling?
So, I mean, here it shows it happening within like an hour. I mean, because it's like 1 minute plus 6 minutes plus 5 or 6 plus 30 to 40.
But like how much time between like it releasing the granules?
It can start over pretty much automatically. So, you know, this CTL is going to go through this process. Yep, Jazz.
I imagine if the CTL only has a finite amount of granules within it,
True.
it can only have so many interactions with
True.
the respective virus infected cell, correct?
Correct.
Is
That.
there a maximum?
How would you measure that?
Oh, I guess
It's basically like almost impossible to measure that. So it can kill again. I don't know that we have fine enough detail to really be able to answer that question. I don't think that that's something that's really probably measurable. So like I said, there are a bunch of things in those granules, and we need to think about what's in the granules. And I'm telling you about 2 different members of the granules, 2 different things that are in the granule. One of the things that's in the granule is something called perforin. It's a protein called perforin. When you hear the word perforin, what does it sound like or make you think about? What does perforin sound like?
Perfume.
Okay, perfume. What else does perforin sound like? Perforate. You heard the word perforate before? What's a perforate mean? To make holes, right? To poke holes in. Perforin pokes holes in cells. It basically perforates the cells. It's a protein that perforates the cells. And so you can see here's our granule. It's got perforin in it, and that perforin gets released, and it basically just makes holes in the membrane of that target cell. And so now sort of general osmotic lysis can happen in the target cell. And so you can see these are actually some perforin pores on the surface of a cell that has been killed by a CTL. So you can see perforin leading to these little pores. So that's going to lead to cell death. That's, again, I'm holding myself back about random fun facts. That's the answer here. So perforin is one thing that's in the granule. Perforin's pretty good because, as you can see, it makes a pore in the hole in the cell membrane. And so that is in and of itself pretty bad for the cell. But the perforin pore has a second job as well. And the other thing that the perforin pore can do is that the perforin pore can let other stuff into the cell. So some of the other things that are in the granule are able to get into that target cell through the pore. So perforin is both like destroying the membrane, but also letting in other toxic stuff. And the big other toxic thing that I'm going to tell you about that it lets into the cell is a protein called granzyme B. So if you notice the name granzyme B, it ends in zyme. So what does that make you think about it? It's an enzyme. So it's some kind of enzyme. Guess what? It's an enzyme that was in a granule. So it's called granzyme because it was an enzyme in a granule. And it was the second one they found, so it's B. So we have granzyme B. So you can see the cytotoxic granule at the top has granzyme B and perforin. You can see we get this little perforin pore. It also is going to let granzyme B into the cell. And granzyme B can start all that same mitochondria apoptosis stuff. And it can also be like, that's too long. I'm just going to turn on caspase-1 myself directly. And so it can, through multiple mechanisms, induce apoptosis, but one of them is it actually, by itself, just turns on the bottom of the pathway. It just turns on the caspase-3 and the cell's done. We don't have to go through all these other steps. It does actually help the other steps happen too, but it just also like skips to the end. So the cell gets very dead as a result of getting granzyme B as well as perforin. There are other things in the granule as well, though I'm not going to go into great detail about them. What is this doing? And so you can see, here's my CTL. It is able to kill an infected cell by fast ligand interactions, as well as perforin and granzyme interactions. And so you can see perforin and granzyme. You can see fast ligand. CD8 T cells are also very famous for making the cytokine interferon gamma that can play some important roles in adaptive immune responses. And so, yes, there's other stuff listed on this slide. But when I think about a CD8 T cell, I think of perforin and granzyme. I think of fast ligand. I think of interferon gamma as sort of the big things that the CTL is using in order to kill a cell that is presenting on MHC class 1, which is going to likely be a virus-infected cell. So that's the CD8 T cell story. That's CD8 T cells doing their effector function. Would I love to like go on and on and on about it because I wrote a dissertation about it? Yes, but we're not going to. Instead, we're now going to do the other side and think about the CD4 T cell. So again, these CD4 T cells start out as naive CD4 T cells, and they can get activated through signal 1, signal 2. They need some IL-2. They will do some clonal expansion, so they will divide and divide and divide, and they will also differentiate and gain some new function. If you look at some places in like some random sites, you might even see this on a couple slides because I didn't edit all through what the book says. Sometimes it talks about IL-2 as being particularly important for CD8s. As far as I'm concerned, you can just say IL-2 all T cells. If you see like IL-2 being special for one kind of T cell or not, ignore that. IL-2 is for everybody. And you can see that in this image as well. You can see that those CD4 T cells are going to expand, and they're going to then get their effector function. But their effector function is not going to be being a killer or cytotoxic T cell. It is being a helper T cell. And sometimes when we talk about helper T cells, we will talk about TH cells. So you can see there's a T with a little subscript H. So if you see something that says TH like that, it generally means an activated CD4 T cell, a helper CD4 T cell. If I think really broadly about CD4 T cells, the effector function that the CD4 T cell gains is that it has the ability to make cytokines to help other cells. And in some ways, this is very similar to what you saw with the CD8 T cell, because once our CD4 cell becomes activated, it's actually going to have a bunch of granules full of cytokines now. This doesn't show it as nicely, that there's granules full of cytokines just chilling, but they are. And when that CD4 T cell interacts with some other cell, we're going to secrete those cytokines right at the other cell through that little, inside that little integrin wall. So you can see the cell-cell contact, and you can see that the cytokines are sort of secreted right at the other cell. And so that sort of secretion part of this looks really similar. What you will notice that I have not told you is what cytokine we're talking about. I've just said the cytokine. And I've just kind of said the other cell. And the reason for that is that there are actually different kinds of helper T cells. There are different types of activated CD4 cells that make different cytokines. So when a T cell starts, it starts as a naive CD4 T cell. In the case of CD4 T cells, so it starts as a naive CD4 T cell, it gets activated. What's not really shown here is when it gets activated, it also becomes a particular kind of helper T cell. So it's all helper T cells are not the same, there's different subtypes. And so it kind of picks, well, it picks a subtype, we'll say, at this point. It picks which subtype it's going to be. So it starts out as just kind of could be anything, and is going to pick a subtype because of sort of what events happen to it in its activation. Sometimes we talk about the naive T cell or the T cell before it has picked a subtype as a TH0. And so cells basically start out as TH0s, and they have to pick what kind of activated CD4 cell, or what effector function they want, what cytokine they want to produce out of the different choices. I am anthropomorphizing this in that they are making decisions in a way that is not entirely true, but that's okay. And the way that this really is working is that when our CD4 cell is getting activated, when it's starting as a naive CD4 T cell or a TH0 cell, it's going to get activated by signal 1 and signal 2. but it's also likely going to receive some cytokines. This is in addition to the IL-2 that we've talked about. It's going to receive some other kind of cytokine. And that other kind of cytokine is going to tell the cell, hey, you should grow up to be a Th1. You should grow up to be a Th2. It's going to tell it which subtype it should grow up to be. So basically, whatever the experience is like when that cell is first activated will push that cell down a particular type of subtype. We'll push it down like a particular track. So that original cytokine, as you can see, is coming from the APC, often is coming from the dendritic cell. And so this is really related to what was going on with the innate immune response. What cytokines did the dendritic cell to start making? And thus, what direction is the dendritic cell going to push T cells? It turns out, I'm doing these slides in a slightly different order because it's making more sense in my brain right now. It turns out that these different kinds of CD4 T cells, these different subsets of CD4 T cells, are useful in different situations. As you will see, one of them is like particularly good at helping you make a response to fungi. One of them is really good at helping you make a response to extracellular bacteria. One of them is really good if you have like an intracellular thing or a virus. There are different types, these different types of activated T cells are useful in different situations. And so the idea, which will come up again on a later slide, is that the dendritic cell, the innate immune cell, will have some idea of what kind of pathogen it is, based on what PRRs got triggered. And so the dendritic cell might be like, oh, I got a fungus here. There was a fungus PRR. So a T cell, please grow up to be the kind of T cell that's good at fighting fungus. And so there's this coordination, this bridge, where the innate immune cell is activated in a particular way by a particular PRR, makes a particular type of cytokine, and that pushes the T cell to be activated to become the type of T cell that you want in this situation. And so here you can sort of see that working out, where we have different kinds of microbes hitting different PRRs. That changes what cytokines are made by the dendritic cell, and that changes what kind of T cell will be activated by that. This is the thing that is a little wrong about this figure, is this shows like this one dendritic cell is making many different T cell types, and that is not the case. Each dendritic cell is pretty much going to push any T cell in one direction. It's just showing all of them on this. But like there might be one dendritic cell that like just starts making IL-12, and so any T cell that comes around it is going to become a Th1 cell, or so on through some of these different things. Yep.
So during clonal expansion, when the naive T cell gets exposed to something like this one specific cytokine, all of those progenitor clonal expanded cells will be that one T cell.
Yep, they will all become that one T cell subset because they're all getting IL-12, or they're all getting whatever cytokine is being produced at that time. Yeah, Jazz.
And T cells also produce cytokines. Is there a feedback?
We're getting there. So this slide, and yeah, so this is one of my future slides, but I have to go back to a past one in a little bit. This is one view from another textbook of different choices that the CD4 T cell could make. So you can see we have our naive CD4 T cell, and we've got all sorts of different types of cell that CD4 T cell could become, different subsets. And really, the way that these cells differ is what cytokine they make. And so they all make a different kind of cytokine that's useful in a different situation. So if, for example, we want to get rid of a fungal infection, there are particular cytokines that are useful to make. So basically we see that T cell kind of getting specialized to make the right cytokine for whatever that infection is. Now, if this shows, what is this? 1, 2, 3, 4, 5, 6, 7 subtypes. You can find literature that has more than 7. You can find literature that has fewer than 7. Sometimes you can find literature that says this is the T cell that responds to gram-positive bacteria with flagella on your right pinky finger on Tuesdays. Sometimes people get a little specific here. And so when I think about this, and when I think about my helper T cell subsets, I kind of think about there being 3 pieces to this whole thing. So it's sort of like sometimes I don't quite buy it when people try to say, oh, there's that one in your pinky on Tuesdays and only your left pinky, right? I'm like, no, like you have to meet these criteria for me to say you're a real subset. And those criteria are, we have to know what cytokine is coming from the antigen presenting cell. So that's listed here as the polarizing cytokine. So what cytokine tells the naive T cell to become this type of cell? We have to know when that happens. We need to know a specific transcription factor that the T cell turns on. So basically, when the naive T cell receives its cytokine, it's going to start making a particular transcription factor. And the idea is that transcription factor is going to tell the cell how to do all the things it needs to do for that type. So it's like there's a transcription factor that is like, okay, if I do this transcription factor, it makes me do all the things for my subset. So we need to know what's the polarizing cytokine. We need to know what's the transcription factor. Here it's listed as a master gene regulator, but I might just also call it a transcription factor. And as a result, this T cell is now going to make some kind of cytokine. And so we also need to know what is the special thing that this T cell does. What is the special cytokine that this T cell does? And if you can't tell me all 3 of those things, I don't believe you that your thing is a subset. I'm still only going to tell you about some of the ones that are real. I'm still not telling you, you will not hear about all 7 that are on this slide. In fact, you are really going to only hear about the 5 that are on this slide from your textbook. Okay? And so what you can notice is that this table for all of these cytokine, or for all of these situations, tells you what is the polarizing cytokine. What is the cytokine that induces differentiation? So what cytokine does the naive cell receive that tells it to grow up into that lineage? What is the transcription factor? And what is the cytokine that is produced? And we can also think about what is the sort of useful thing that's happening. What is the situation where that T cell is useful? Why do you want that T cell? What is that T cell good at? So we're going to talk through this in detail, but this is sort of an overview of what we're going to be talking through. So when this was first understood, people started to say, okay, there's 2 kinds of helper T cells. Again, we now know there's more than 2 kinds, but originally they thought there was only 2. And they called them Th1 and Th2. Helper T cell kind 1, helper T cell kind 2. And what they thought happened with Th1 and Th2 cells was that if the dendritic cell made IL-12, when it saw the naive T cell would grow up to become a Th1 cell. And when you grow up to become a Th1 cell, you make a transcription factor called TBET. And you make, in particular, the cytokine interferon gamma. Alternatively, if you are a Th2 cell, you receive IL-4 during differentiation from that dendritic cell. That makes you make the transcription factor GATA3, and then you yourself as that T cells start making a few cytokines like IL-4, IL-5, and also IL-13. So if we have our Th1s and our Th2s, the polarizing cytokine for Th1 is IL-12. The polarizing cytokine for Th2 is classically thought of as IL-4. The transcription factor for Th1 is TBAT. The transcription factor for Th2 is GATA3. And the cytokine that is produced by Th1 is interferon gamma, while for IL-4 for Th2 it's IL-4, IL-5. It also can be IL-13. Sometimes students are a little bit confused when they look at this because they're like, wait a minute, for Th2s, you start with IL-4 and you get IL-4. That seems dumb. And so we also think that there might be this other cytokine called IL-33. That is actually important here as well, though that's newer information. But at least classically, everyone, it's always, you start with IL-4, you get IL-4. But if that confuses you, I'm going to be writing more. I would take a picture at the end of class. So that is what we usually think of for Th1 and Th2 cells. And so here you can kind of see some general ideas of the Th1 cell. And so you can see here's our dendritic cell. It's making IL-12 to give to that naive T cell. I don't know why it's shown here and not there, but I can't draw, so what can I say? Here you can also see the dendritic cell is actually giving the cell IL-12. That cell starts to make TBAT. When the cell starts making TBAT, now this T cell is called a Th1 cell, and it's making this product of interferon gamma. And interferon gamma is pretty important. Interferon gamma helps MHC presentation, so it really helps, especially class 1 presentation. So it says, hey, do make more class 1. Make sure you're presenting. It turns on CD8 T cells. It helps the CD8 T cell be better. And it also is particularly good at helping to initiate any response to an intracellular pathogen. So we're seeing really good MHC class 1. We're going to turn on CD8s. We're generally going to be good against intracellular pathogens. The other big thing that we think about when we think about the Th1 cell is that that interferon gamma coming from the Th1 cell also really activates macrophages. And so here you can see a Th1 cell interacting with a macrophage, giving that macrophage some interferon gamma. And as a result, that macrophage gets really good at killing things in its phagosome. That macrophage is now going to be super spectacular at killing anything in its phagosome. And so in a lot of ways, people often, classically, originally, people thought of the Th1s as like the inflammatory T cells. Not really true anymore, but you're gonna take a picture later because I'm gonna write more stuff. It's okay. I will tell you guys at the end. I'm gonna be doing this until for the rest of class today. At the end of class, take your picture. Yes.
Is there a specific location that we should think about this happening, or
I mean, so the T cell is gonna interact with the dendritic cell in the lymph node, but then it could go anywhere.
so it like activates a macrophage and then that macrophage can
Yeah, and so this could be, well, so no, it's probably going to activate the macrophage in a tissue, but it's wherever that T cell has gone, wherever, you know, because it's activated at this point. It's not in the lymph node anymore. Okay. It's a grown-up T cell, and it's going to be wherever it is, find a macrophage and activate that macrophage. And so it sort of depends on what's the infection, what's the microbe we're talking about, where this might be important. Alternatively, Th2 cells are turned on when we make IL-4. So here you can see my dendritic cell making IL-4 for the helper T cell. Again, I don't know why it's in the wrong spot, but that's fine. Now the Th2 cell will start making this transcription factor GATA3, and that tells that cell how to do things like make IL-4, IL-5, and IL-13. These particular cytokines are good for different functions. So you would want a Th2 cell in a different situation. Th2 cells and the Th2 cytokines activate mast cells, and they can also help B cells, although these are not the real stars for B cells, so I'm not even going to write it on here. I'm going to say activate mast cells is going to be the big thing. And so if you think about it, the mast cell is secreting toxic stuff. A mast cell is really good for for something that's extracellular, that's outside of cells, that is not in a macrophage. Macrophage can't eat it, so we need to use some other mechanism to kill it. So you can see here that in this case, the microbe it's showing you is a parasitic worm that is not getting phagocytosed. It's extracellular, and we want to secrete stuff at it to try to kill that extracellular parasite. And so Th2 cells tend to be very good at dealing with extracellular parasites. Th2 responses, the other thing that is actually really important with the Th2 response is you do get some IgE class switching. And so again, you're going to get IgE production in particular with Th2 cells as well. So you're going to get some IgE, which again was really important for activating mast cells. So we're going to get a good IgE response. We're going to get a good mast cell response. All of our different helper T cell subsets can go wrong. Sad. So many things in immunology can go wrong. And they each are associated with different things when they go wrong. Th2 cells are the reasons for many, many, many of your allergies. Your asthma, your respiratory allergies. Th2 cells gone awry cause me a lot of problems in my life, and mostly I like to say mean things about Th2 cells because they give me all of my allergies. Th1 cells can be involved in some inflammatory and autoimmune diseases, but that's stuff we'll think about later. But honestly, when I think of Th2 cells, I think of allergies most of the time. And so here you can see some of the details of this. We can see we get some IgE production with a Th2 cell. You can see we're going to be activating mast cells. We are also potentially activating eosinophils. We also do things like have a lot of mucus secretion and peristalsis. You have this big worm that you can't phagocytose. One thing that could help get rid of it is to get rid of stuff, is to basically start throwing up and having diarrhea and having lots of mucus come out your nose and basically trying to get stuff out. And so in fact, mucus secretion and peristalsis to just expel whatever you can often will happen with these same cytokines. And again, you can think of that in certain allergic reactions where you're really expelling. And so all of those things are sort of the function of a Th2 cell when that cell is activated. And this is particularly useful for extracellular parasites like the parasitic worms that are shown here and on the previous slide. So there's our activated mast cell. It's secreting stuff to kill extracellular parasites. And so this is sort of the original classic view of thinking about Th1 versus Th2 cells. So you can see that our dendritic cell is going to see a different kind of microbe with a different kind of PRR. It's going to make a different kind of cytokine. And so when that naive T cell sees signal 1, signal 2, and whatever cytokine it's getting, it's going to be polarized in a different direction, either say Th1 or Th2, So you can see now it becomes a Th1 or Th2 stuff, Th1 or Th2 cell, and then it can do either the Th1 job or the Th2 job. And what's really great is that that job is actually the best possible job for whatever that original microbe was that was sensed. The other thing that comes up here that is really key, two other things that come up here that are really key. One is that different cytokines made by the helper T cell will influence what kind of class switching B cells do. So we haven't really talked about B cells in the periphery yet. That's going to be an after the exam problem. We'll deal with them later. But when those B cells are in the periphery, they might switch to something other than IgM or IgD. And the way that those B cells decide which other cytokines to switch to, or which other class to switch to, whether they make an IgG type or IgA or IgE, is based on what cytokine they get from T cells. So basically, whatever kind of T cell you get is going to dictate what kind of antibodies you get. It's also going to dictate, are mast cells active, are macrophages active, things like that. Sometimes I hear people talk about the CD4 T cell as like the quarterback of the immune system. It's the one that's going to decide what direction we're going to throw it in and which direction we're going to have everything else go in. When I hear that, I say, okay, that's fine, but the dendritic cell is the coach that told the quarterback what to do. And so you can see that sort of whichever type of CD4 T cell we're getting is going to bias the rest of the immune response in a particular direction, which also includes what kind of cytokine we get and what kind of antibody class switch we have. But this is the other thing I thought I was going to be telling you on the last slide. There's one more piece to this, and this relates back to David's question. So David asked a question about, if you have one dendritic cell that's turning on a few different, like one T cell has lots of progeny, there's clonal expansion. David was like, are they all going to be Th1? And I said, yeah, because they're all going to get IL-12 because they're all in the same place getting IL-12. But it's actually more than that. Not only does IL-12 turn on TBAT and thus have interferon gamma turned on, IL-12 also directly inhibits GATA3. So not only does that cytokine say you should become a Th1 cell, it also actively inhibits a Th2 production. So really, T cells are deciding, I am Th1 or Th2. There is a feedback loop that once you become a Th1 and start making TBAT, you make Th1 cytokines, and you specifically inhibit Th2 cytokines. So basically, you have a positive feedback loop to do more Th1 things, and you have a negative feedback loop that's inhibiting any Th2 situations. And so often, different immune responses that happen in different situations tend to be super Th1 biased or super Th2 biased. You tend to make a lot of Th1s or Th2s in situations, and there's sort of feedback, both positive and negative, pushing everything in that direction. As we'll talk about when we get to allergy later, genetically, some people are predisposed that their body just likes to go Th2, and then they have a million allergies. Whereas other people are maybe predisposed that they tend to push towards Th1 more often. And so, basically there is this feedback where once you're doing one type, you're really committed to going on that one type. And you're going to see most of the response happening to and being that one particular type. So this is where we were when I started graduate school. We had Th1 and Th2. It's all very easy. And then people kept finding more T cell subsets. The third one they found, I'm not telling you about right now for reasons. We'll get there. So I'm actually skipping ahead to the fourth one they told us they found. And you might look at this one and be annoyed and be like, immunologists, seriously? Do you really not know how to count? We remember with that complement business you were bad at counting, but really? Because this next subset I want to tell you about is called the Th17 cell. But you will eventually like the name Th17. So Th17s were sort of one of the next big subtypes that were described. The reason why Th17 cells are called Th17 cells is because they make IL-17. So they got named Th17 because they make IL-17. And so you can see that here. Th17 cells are particularly important, know to be Th17 cells when they start making a transcription factor called ROR gamma T. So you can see that transcription factor here. And when we polarize Th17 cells, couple key things. One, you will find different answers in different sources for this one. There are two things I want you to care about with this, okay? With a Th17 cell, one thing that's really important is you need a combo of two cytokines for Th17 cells, okay? One of the cytokines that you always need is called TGF-beta. So you always have to have TGF-beta, but not just TGF-beta. You also have to have, as this image says, IL-6. You can see it also might be IL-23. There are other places where you'll find other cytokines as well. The thing that all of the cytokines here have in common is it's an inflammatory cytokine. So basically you need TGF-beta plus an inflammatory cytokine. If you were, say, answering a question about this and what the polarizing cytokine was for Th17 cells, I would want you to give me TGF-beta and any inflammatory cytokine that you find in any other source. I will mark anything that is in any of the slides or in the textbooks correct, as long as it's TGF-beta plus one of those other things. So here I'm telling you IL-6, because in a lot of ways IL-6 makes a lot of sense here. But if you see something else, if you see IL-23 or something in another figure and that's what you learn, as long as you give me TGF-beta plus that, I'm marking it correct. And what you can see here is what the important role of IL-17 is. So IL-17 is particularly good at activating neutrophils. It is very good for extracellular bacteria and any fungi. So if you have some IL-17, you're going to have a lot of neutrophils and you're going to be really good at responding to extracellular bacteria and fungi. And this is because the Th17 cells are making IL-17. Th17 cells, again, are involved with a number of different autoimmune diseases, inflammatory diseases we can see later. The next big helper T cell subset that was described was called the TFH cell. And the TFH cell, again, you are going to see different cytokines in different sources. If you give me a cytokine that is listed in any slide, in the textbook, whatever, I'm going to mark it as right. So I'm just going to put some here based on this slide, but if you happen to give me one that you studied from somewhere else, it's okay. The key thing with a follicular helper or a TFH cell is it's making the transcription factor BCL6. You can see that the TFH cell, for example, might make a cytokine or might get polarized with a cytokine called IL-21 or also IL-6. And generally, I think about it as making IL-21. And Emily asked when we were talking about Th1 cells about particular locations for Th1 cells. This is the only one of these types of cells where there's a specific location. So this one does have one place where it usually works. And the one place where it usually works is related to the one function it has. So the one place where this cell usually works is in a part of a secondary lymphoid organ called a follicle. So the TFH cell works in the follicle in the secondary lymphoid organ. In fact, the name TFH stands for T follicular helper because it's helping in the follicle. So it's the T follicular helper. And as you will see later, the follicle is where B cells get turned on. So these are the B cell helpers. These are the ones who turn on B cells. So these cells are really important for turning on our B cell in the follicle. And again, more B cell details will happen later. So those are kind of sort of some big four subsets. But there is a fifth one that I want to tell you about. The fifth one was sort of discovered through a little bit of a circuitous route. And has a couple of things about it that are kind of interesting. And so that's why I'm setting it up separately from the rest. It was technically the third T-cell helper subset that was described. Once upon a time, it was called Th3, but no one ever calls it Th3 anymore. And so I'm going to spend the rest of today telling you about those cells, but also know that the paper that you're reading for Monday involves these exact same cells, and we'll be hearing more about these cells coming up. And if you remember, I told you last time that there were certain words that I learned when I was a student that like immunologists should not ever say. You get struck by lightning. Lightning's going to strike me down if I say them. And I said one of them last time, which was danger. We'll say the other one now. So there was this hypothesis for a long time that there were T cells who could turn off other cells. They could turn off other T cells. They were like the off cells. So whenever other T cells, macrophages, B cells, anybody was sort of doing wrong, these other T cells could turn them off. And the idea was that they were known as suppressor T cells. Suppressor is the other word that's going to get lightning to strike me. So there's this whole literature of suppressor T cells, suppressor T cells, suppressor T cells. And everybody's really into suppressor T cells. They actually figured out this gene that was really important with suppressor T cells. It's called IJ. And then they did some genetic studies, and IJ didn't exist. There was no gene in the place for the IJ gene. And basically what happened is the field was like, what? Suppressor T cells? I didn't believe that. I thought that was fake all along. I don't know what you all were talking about. I never believed that. Suppressor became like the dirtiest word in all of immunology. Nobody would talk about suppressor T cells. And everyone was like, that is fake. There is no such thing as an off T cell. No, 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 no. In 1995, somebody did some pretty cool experiments to find and sort of describe this T cell that seemed like it was turning off other cells. But the word suppressor at that point was like tainted. We could not have the word suppressor. You will notice, basically, immunologists never use the word suppress, even now. We picked another word. And when we say it, we mean suppress. But we don't say suppress ever. And so what we like to say, when we want to say suppress, is regulate. And so we started saying, no, there are no suppressor T cells. There are regulatory T cells. In this paper in 1995, someone first really had some good evidence. Sakaguchi had some good evidence for the existence of regulatory T cells. But he didn't have good markers. He sort of didn't understand some of the proteins. Some of the proteins he had on them were kind of confusing. And then in 2003, people actually figured out some good markers and some good proteins and could really prove that regulatory T cells were a thing. Sakaguchi, in fact, did some of that work himself, as did others. Sakaguchi and the other two, the others that I just mentioned, won the Nobel Prize this year for discovering all of this. And so this is our final set of cells. Oh, see, look, T follicular helpers helping B cells. I forgot that I was going to tell you that, but yeah, that's what they do. So the other cell type is a cell type called the regulatory T cell, or a Treg. The idea, if you ever hear me or if you see in a paper or see someone talk about regulation, they often mean suppression, or they often mean turning off. We just can't use the word suppression. We have to use the word, we can't use it anymore. Nope. And so we have a lot of things have been described about Tregs, regulatory T cells. One thing that's sort of interesting about regulatory T cells is that there are still pieces of them that are not well worked out. I actually found it fascinating when I was looking at the paper that I'm having you guys discuss on Monday, that there are some things in there that are like directly contradicting your book. Because in fact, some of the details of certain parts of Tregs have changed and have been uncertain a bit in a couple of places. But the idea is that the Treg is going to be able to turn off some other cell type. Here it's turning off some kind of other cell type that's a CD4, but it could be many other types of cells. This turning off, and so we've got our Treg. It's turning off other cells. And it is doing that by making a cytokine called IL-10, at least for our purposes. Though there are debates about other ways that Tregs can turn off cells. For our simple purposes right now, I'm saying IL-10. One of the things that is a big debate, that again, I'm not going to get super into, though it will come up with the paper, is there's a question of, is the thing that Tregs use to suppress a secreted cytokine, or is it actually a protein so the cells have to touch? And so there's actually a whole figure in that paper about, do the cells have to touch or not, which we'll see later. We're just going to say it's secreted IL-10, and I'm not going to we'll deal with the rest of that rabbit hole after the exam. Right now we're just going to say suppression happens with IL-10. Here you can see IL-10. Here you can see IL-10. The key thing for Tregs is that they have a specific transcription factor. That transcription factor is called FOXP3. And in fact, the reason why sort of Tregs became real in 2003 is because people found FOXP3. I actually was listening to a talk in grad school of the first ever description of FOXP3 and what it did. I was a first-year grad student. I had no clue what was going on in that talk. I remember being like, Fox, who? And now I'm like, oh, that was actually the first description of FOXP3. Wish my first-year grad student self had better clue what was going on. And the thing that's interesting about Tregs is that Tregs can be made in two ways. So there are two different ways you can make Tregs. Sometimes people kind of would make them as two separate columns here. I'm just going to kind of mention one type here, and then I'll give you the other type on the next slide. Okay? So there are two ways you can make Tregs. One way you can make Tregs is if a naive T cell sees TGF beta without an inflammatory cytokine. So the TH17s are TGF beta with an inflammatory cytokine. If a naive T cell sees TGF beta alone, it becomes a Treg and starts turning off other things. So you can kind of think that it's like, okay, it's TGF beta, I'm going to get activated. Should I get activated towards inflammation, or should I get activated towards being chill and turning everyone off? And so you can see sort of that Treg outcome here. But the other thing is that some Tregs come out of the thymus as Tregs. They don't actually have to get activated in the periphery. So these Tregs that I have on the board are Tregs that were induced in the periphery. Sometimes people call them inducible Tregs, or there are a bunch of names. Sometimes people call them inducible. I think some people have called them adaptive Tregs. Whatever. It's getting activated just like any other helper T cell subset. But some cells come out of the thymus already Tregs. They already have been told, you're going down the Treg route. They never have a chance to pick the other ones. They're never a naive T cell. They're just come straight out of the thymus, already committed to being a Treg. And when those cells are coming out of the thymus, already being a Treg, you can remember that when we talked about the thymus, we talked about a situation of no or low signal, where we have the death by neglect. We have sort of this lower intermediate signal, and then we have a high signal. Remember, high signal was deletion, while low intermediate was positive selection. Positive selection, aka you become a T cell or become a naive T cell. Well, this was negative selection. However, if there are some cells that have kind of a high intermediate, not really high enough that we're like, I gotta kill you, but high enough that they're dangerous and we don't really want to positively select them. So this is actually a fourth outcome. Not the absolute strongest signal, but like the next strongest signal. So still kind of dangerous cells that maybe shouldn't go into the periphery as activated T cells. Those T cells, which sort of have this middle affinity, as you can see here, I have it listed as high intermediate, start turning on FOXP3 in the thymus. So it's not that they got TGF beta, it's like they got thymus selection. They made FOXP3 and became Tregs and started making IL-10. So the other outcome in the thymus is that cells can be induced to become Tregs. And so here, I showed you this before, but I actually had put a white box that said that over Treg production, and so you can see that these cells sort of that are on the higher middle end are going to become those Tregs. Regulatory T cells are able to regulate, aka suppress other cells in a bunch of ways. One of them is that they make cytokines. This says TGF beta, I'm saying IL-10. Where's going to be IL-10? It's just going to be IL-10 because I'm trying to like simplify. There may be some sort of contacts that may happen, but another really big thing is that Tregs love them some IL-2. They are IL-2 hogs. They have CD25, they have that high affinity IL-2 receptor on their surface. And so one thing they can also do is just take all the IL-2 to deprive anyone else of having any IL-2. So a lot of this is about taking IL-2, using suppressive cytokines like IL-10 or other mechanisms. And we now know that all of these different T cell subsets actually have that same feedback loop that I told you about before. It's not just like Th1s have positive feedback for more Th1 and negative feedback for Th2. It's actually Th1s have positive feedback for Th1 and negative feedbacks for all the other ones. And you can see the same kind of interaction amongst all of these different T cell subsets. And there's a summary slide, and I'm done. I will see you for the review session. Now you can take your pictures and let me know what questions you have before the exam.